Hello everybody. So this is my presentation on muscle contraction, and this one was quite a doozy, so I'll just get right into it. So what a muscle contraction is when the muscles shorten and they generate force and movement. The structure of muscle cells are muscle fibers, which are long cylindrical cells that make up muscles. And then we have the myofibril, which is a long cylindrical organelle that runs parallel within the muscle fiber. And those contain the sarcomeres, which they're all found inside the muscle fibers. And the filaments are what myofibrils are composed of. And the sarcomere is the smallest contractile portion. And the filaments that are found within the sarcomere is the thin filament, which is primarily made of actin, and then the thick filament, which is primarily made of myosin. And so that gets into the sliding filament process. Okay, so with the sliding filament theory, it gets broken down into 6 different steps. The first one is starting the contraction, and that is when the nerve signal triggers the release of calcium. So a nerve impulse from a motor neuron travels to the muscle fiber to give a message from the brain telling the muscle to move. And this message makes calcium get released into the muscle that's being contracted. And then the second step is that the calcium expands the binding sites on actin, and it exposes the actin sites. So calcium binds to a regulatory protein called troponin, and it causes a shape change to move the tropomyosin out of the way. So in turn, this exposes the binding sites for the myosin. And usually the troponin and the tropomyosin is blocking the binding sites on the actin. So they'll look kind of like this, where they're not really connected when it's not being contracted. So the calcium helps open up for opportunity to bind together and start the contraction. And then the third one is the myosin binds to the actin, and so that's the cross bridge formation. And in this step, the myosin heads can now attach to the binding sites on actin, forming cross bridges. So we got those regulatory proteins that were in the way, they cleared the way. So as you can see in this picture, the actin usually looks like that, and it's usually blocked off, but the myosin heads now open up, and then they'll be ready to form a bridge in that gap. So they'll be connected. And then the fourth step is that the myosin pulls the actin, so it's a power stroke. So the myosin head pivots after the attachment, and it pulls the actin filament towards the center of the sarcomere, which shortens the muscle. So as you can see, the myosin is connected by an M line that's running straight down the middle. I'm so sorry, I think it was labeled as a Z line in the beginning. And the Z line is where the actin or the thin filament is connected. So they pull together, and the myosin pulls the actin towards this midline, towards the M line, so it'll shorten and contract. And then the fifth step is that the myosin releases and resets with the help of ATP. So the ATP molecules bind to the myosin head here, and then that causes the myosin to detach from the actin, and the ATP is then broken down into ADP and phosphate, which recocks the myosin head to be ready for another contraction. So the sixth step in the sliding filament theory is that the cycle repeats and shortening the muscle. So as long as calcium and ATP are present, the cycle of grabbing continues. So the muscle keeps on contracting. So they go through this whole process all over again. It's really fascinating. I never really knew how complex the body is, and just something so simple as a muscle contracting, there is a lot that goes into it. So the role of calcium and ATP is that calcium binds to the regulatory proteins on the filaments and exposes the myosin binding sites, which allows the myosin heads on the thick filaments to attach. So again, attachment over here. And then the ATP provides the energy for the sliding process, which continues as long as calcium is present. So it's just a continuous cycle. Thank you so much for watching my presentation. I hope you learned something, and I'm really excited to see everyone else's presentations and learn even more about this. Thank you. Bye-bye.